Polygenic inheritance. It is the inheritance in which some traits are controlled by three or more genes, that is, multiple alleles, example human skin color, human height, etc. Polygenic inheritance also takes into account the influence of environment. In a polygenic trait, phenotype reflects the contribution of each allele, that is, the effect of each allele is additive. Human skin color. Assume that three genes A, B, and C control human skin color. Dominant forms A, B, and C responsible for dark skin color. Recessive forms a, b, c responsible for light skin color. Genotype with all dominant alleles will give the darkest skin color. Genotype with all recessive alleles lightest skin color, and genotypes with 3 dominant alleles and 3 recessive alleles will give the intermediate skin color. Thus, number of each type of alleles in a genotype determines the darkness or lightness of the skin. Next, in pleiotropy, we did starch synthesis in a pea plant. Here, starch synthesizes effectively by B gene. Therefore, large starch grains are produced. Small and small b have lesser efficiency in starch synthesis and produce smaller starch grains. It also shows incomplete dominance. That is, if we made effective starch synthesis B with small b, that is a large size seed with a small size seed, we will get an intermediately sized seed, that is B small b. Next, we have sex determination. In a human, there are 46 chromosomes and 23 pairs of chromosomes, which are autosomes and sex chromosomes. Autosomes, it controls the somatic character and it is the first to 22nd pair. Sex chromosomes, on the other hand, it controls the gender of human and it is the 23rd pair. X, it could be X or Y. If it is XX, then it is female, and if it is XY, then it is male. Next, we will go to autosomes and sex chromosomes, which are also known as allosomes. Autosomes are chromosomes other than sex chromosomes. Number of autosomes in male are same in male and female. Sex chromosomes X and Y are the chromosomes which are involved in sex determination. Henking in 1891 studied spermatogenesis in some insects and discovered X chromosome. Chromosomal theory of inheritance, proposed by Walter Sutton and Theodore Boveri in 1902. It states that the pairing and separation of a pair of chromosomes lead to segregation of a pair of factors they carried. Sutton united chromosomal segregation with Mendelian principle and called it the chromosomal theory of inheritance. The important statement of chromosomal theory: chromosomes are vehicles of heredity. They are transmitted from parents to offspring, that is, they are immortal. Two identical chromosomes with similar genes form a homologous pair. Homologous pairs segregate during gamete formation during anaphase 1. Independent pairs segregate independently of each other. Chromosomes are mutable. Genes are present on chromosomes, hence they show similar behaviors. Next, we move on to Morgan's experiment. Thomas Hunt Morgan worked with fruit flies that are Drosophila melanogaster to prove chromosomal theory of inheritance. Drosophila is a suitable material for genetic study because it breeds very quickly, short generation time, that is, life cycle is from 12 to 14 days. Breeding can be done throughout the year, hundreds of progeny per mating. They can grow on simple synthetic medium Male and female fruit flies are easily distinguishable. It has many hereditary variations that can be seen with low microscopes. Linkage and recombination. Linkage. Linkage is the physical association of two or more genes on a chromosome. Linked genes do not show independent assortment, whereas recombination is the generation of non-parental gene combination. Morgan carried several dihybrid crosses in Drosophila to study sex-linked genes. The example 1. A recessive female was taken which was yellow-bodied, white-eyed female, small y, small w, small w, and a dominant male was taken which was brown-bodied, red-eyed male, wild type, and y plus y plus w plus w plus. In the next cross, a recessive female white-eyed and miniature-winged, and a dominant male red-eyed and large-winged wild-type male was taken. Here Morgan intercrossed their F1 progeny. He found that the two genes did not segregate independently and the F2 ratio deviated from 9:3:3:1 ratio. Genes were located on the X chromosome. When two genes were situated on same combination, it was much higher than the non-parental type. This was due to linkage. The genes white and yellow were highly or tightly linked and showed 1.3% recombination. White and miniature winged 37.2% recombination, which were loosely linked. Tightly linked genes show low recombination, while loosely linked show high recombination. Here we also note that Alfred Stuart Went used the recombination to find frequency between gene pairs as a measure of the distance between genes and map their position on chromosome. Today, genetic maps are used as starting point in sequencing of genome, as was done in Human Genome Project. Mechanism of sex determination. There are three mechanisms here. The first one, XX and XO mechanism. Here the male is heterogametic, and that is XO, gametes with X chromosome and gametes without X chromosome. Female is homogametic, that is XX, all gametes are with X chromosomes. Example, many insects such as grasshoppers show this type of mechanism. The second mechanism is XX and XY mechanism. Here the male is heterogametic X and Y, and the female is homogametic X only. Example, human and Drosophila. The third mechanism is ZZ and ZW mechanism. Here the male is homogametic ZZ and female is heterogametic ZW. Example of this is birds. So we conclude that XX and XO and XX and XY mechanism show male heterogameity, while ZZ and ZW mechanism shows female heterogameity. Sex determination in humans, that is XX and XY type. Human has 23 pairs of chromosome, 22 pairs of autosome plus 1 sex chromosome. A pair of X chromosome XX is present in female, XY chromosome are present in male. During spermatogenesis, male produce two types of gametes, that is 50% with a X chromosome and 50 with Y chromosome. Females produce only ovum with X chromosome. There is an equal probability of fertilization of ovum with the sperm carrying either X or Y chromosome. The sperm determines whether the offspring is a male or a female. Sex determination in a honeybee. It is based on the number of sets of chromosome an individual receives. An offspring formed from the union of a sperm and an egg develops as a female queen or worker. An unfertilized egg develops as a male drone. It is called parthenogenesis. Therefore, the female are diploid 32 chromosomes and males are haploid 16 chromosomes. This is called as haplodiploid sex determination system. In this system, males produce sperms by mitosis. They have a grandfather and they do not have a father and thus cannot have sons, but they will have a grandfather and can have grandsons. Next, we will go to mutation. Mutation is a sudden heritable change occurring in DNA sequences that result changes in the genotype and phenotype of an organism. Recombination and mutation leads to variation in DNA. There are two types of mutation: frame shift mutation and point mutation. Frame shift mutation is the loss or deletion or gain and insertion or duplication of a DNA segment. Point mutation, mutation due to change in the single base pair of DNA. Example of this is sickle cell anemia. Genes are located on chromosomes, so mutation results in an abnormality. Chromosomal aberrations are seen in cancer cells. Next, we will go to mutagens, that are agents inducing mutation. Physical mutagens. We have UV radiation, alpha, beta, gamma rays, X-ray, etc. Chemical mutagens. We have mustard gas, phenol, formalin, acetic acid, formic, and NH3, etc.